Good evening. My name is Candace and I'm an event manager. On behalf of the staff at Town Hall Seattle and our friends at Third Place Books, it's my pleasure to welcome you to. As we get underway, I would like to acknowledge that our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continued use of the natural resources of their ancestral homeland. So glad to have you join us tonight. This presentation will run about 60 minutes, including Q&A. To submit your questions for the Q&A portion of the event, please enter meet.ps/Shapiro or scan the QR code right now on the screen with your smartphone. We'll drop this link in the chat and remind folks where to go once we get to the Q&A portion of the event. We can't guarantee that we'll be able to address every question, but we'll try to get to as many as we can. You can help us by keeping your questions concise. For those who would like to view the program with closed captions, you can click the CC button in the bottom right corner of the video player. Town Hall is adding new events and podcasts every day. Upcoming events include Jack Gilbert with Sean Gibbons on the promise of the human microbiome, and Meilon K. Han and Albert Rizzo on how our lungs work and how to protect them. Visit our website to join our email list and get the latest updates as more programs are added throughout the season. Town Hall's work is made possible through your support and the support of our sponsors. As part of our Arnold G. Mitulski Science Series, this event is supported by Microsoft. Town Hall is also a member-supported organization, so I want to thank all of our members joining us tonight. If you share in Town Hall's goal of a community energized and empowered by considering questions of civics, science, and culture, please consider supporting us by becoming a member. You'll find membership information on our website. And lastly, you'll want to dig into tonight's topic by purchasing your own copy of the author's book. Please use the link in the chat below to pick up your copy through Third Place Books. Now for this evening's event. Beth Shapiro is a professor of ecology and evolutionary biology at UC Santa Cruz. She has appeared on National Geographic, Discovery, and the BBC, and has written for the Financial Times and Observer. She is the author of the 2015 award-winning book, How to Clone a Mammoth. Carl Zimmer is a science columnist for the New York Times and a frequent contributor to magazines including The Atlantic, National Geographic, and Scientific American. His award-winning books include Life's Edge, The Search for What It Means to Be Alive, and She Has Her Mother's Laugh, The Power, Perversion, and Potentials of Heredity. Shapiro's new book, Life as We Made It, How 50,000 Years of Human Innovation Refined and Redefined Nature, is the subject of tonight's conversation. Please join me in welcoming Beth Shapiro and Carl Zimmer.
Thanks, Candace. And thank you all for joining us. I'm Carl Zimmer, and it's great to be talking with Beth Shapiro. I'm talk to Beth fairly often, just picking up the phone to find out about her latest research or to get her to share her thoughts about some other research in her field. She does some amazingly fascinating work. I'm talked to Beth about, I don't know, fossil horses and Neanderthals, and we were just talking about forensic solving cold cases. So Beth's interests span a huge range, and I guess what ties it all together is DNA, you could say. But it's interesting that we're now talking about your latest book, which is not about fossil horses. It's Life as We Made It, talking about things such as biotechnology, about the potential for altering foods and wildlife and all sorts of things. And so I guess maybe before we kind of dig into some of the big issues that you address in the book, like how well, first let's talk a little bit about your career. How did you end up being a scientist who's off in the Arctic tundra digging up ancient fossils of horses and figuring out what kind of DNA they have in them?
Well, I wasn't expecting that question. Actually, there's a funny thing you say. My book is not about fossil horses. When we were chatting just a minute ago, I mentioned that there was one chapter that I deleted from this book, which was entirely about fossil horses.
But and this if there's another book, just make it a book about fossil horses. Okay, we're
There's
done.
a lot.
All
There's
right.
a lot there, right?
All right.
And present day horses, right?
Absolutely. So
How
everyone
did
can
I?
come back in 2 years, and that'll be the next book talk. But for
2
now,
years? What? Are you kidding? I have a day job too. Anyway, how did I get here? Oh, I guess it's a long and winding route. I grew up in Georgia, the Southeast, and I wanted to be a journalist when I grew up. And I went to the University of Georgia because they had a great journalism school. And after my freshman year, I was fortunate to be able to take this amazing class where we started off on the East Coast of the state and we learned about the accretionary ocean processes, just learning about how the ocean changes the landscape. And then we drove across the country to the West Coast and then back across the country over the top and in the middle. It was 10 weeks and we slept in national parks and we went to archaeological sites and geological sites. And I just fell in love with thinking about how everything changes all the time. Everything is changing. And I thought to myself, people need to know about this. We're about to be in this huge crisis, and we are in this huge crisis, and climate emergency and biodiversity extinction crisis. And we need people who can talk about this in a way that everybody can understand and appreciate, so we can do something to save all of these species that are threatened. So I decided then that I would be a science journalist rather than just a regular journalist. And started taking classes in science, ended up getting sucked in. I had an opportunity to go live and work in Panama and Costa Rica for a while and really enjoyed that. Ended up in the UK to do my PhD, not really knowing what I was going to do. And I met Alan Cooper there, who was a new professor at Oxford from New Zealand. And I was lost. And he said, I don't have any students and I need somebody to go to Siberia. So if you join my lab, then we go to Siberia. And I thought, but that's a great reason to make a life-changing decision, like what to do for your PhD. I joke that it wasn't entirely that. I mean, Alan was working in this new field called ancient DNA, and I had never touched a pipetman. I didn't know anything about molecular biology, but I was interested in geology and ecology. There's my dog, and in the way he joins in as much as possible when I'm doing this kind of thing.
We'll definitely be talking about dogs in this hour.
Hey, kids, shh, please, and my children. So there you go. Please. Sorry.
So,
my husband is meant to be here helping, but he was called away unexpectedly. And so here I am dealing with the dog and the kids in this, and I'm very sorry.
You're doing great.
Anyway, ancient DNA, what it meant was that we could get DNA out of fossils and bones and skin and teeth and hair of animals that used to be alive, even maybe of pieces of plants that were recovered from the sediment. And you could actually trace changes in entire ecosystems and communities directly over time using DNA. And I was hooked. I thought this was an amazing way to really be able to tell the story of how Earth is always changing, our habitats are always changing, and maybe think about what we could do in the future. I'm really sorry about the noise in the background.
Well, as things are settling down there, maybe I can sort of talk a little bit about some of the interesting things that you've been doing in this field. So, for example, I mentioned fossil horses. This is some of the old, hundreds of thousands of years old. This is some of the oldest fossil DNA that anybody's found. You have been involved with working on ancient human remains and our relatives like Neanderthals and other extinct humans and pulling DNA out of and making sense of those fossils. So I mean, I don't know if there are particular other highlights you might want to mention.
Well, you know what, you mentioned horses. So yes. let me throw something at you that is cool and not in the book, and in fact not published at all, but cool. A few years ago, we were working in the Yukon. Well, actually, this is longer than that ago. So you can tell how old a fossil is either by directly dating a bit of the bone using radiocarbon, or if that fossil is below a layer of volcanic ash that settles on the surface like a big blanket. And we knew that there was one part of the Yukon where there were horse remains that were from beneath one of these ash layers, and the volcanic eruption happened around 700 to 750,000 years ago. So we knew that this horse was around 700 or 750,000 years old or slightly older. And until that time, the oldest DNA that people had recovered and authenticated was much less than that, maybe on the order of 60,000 or perhaps a little bit older than that, years old. But there was DNA in this horse bone, and we worked hard on it. This was a collaboration with Eske Willerslev and Denmark and a bunch of other people. And we published a sequence, a genomic sequence of this horse that was just a horse, a normal horse, but 700,000 years old. And then more recently, we've developed some new approaches that are better at getting DNA out of these really old fossils. And we wanted to go back to this horse and try again, just to show that we could get a much better version of his genome. We could learn more about this horse. And the museum curators were like, yeah, no, I don't think so. You already chewed up most of this horse bone, but there's another horse bone, in fact, a skull from this same site, it's the same age. And why don't you try that? Because that one doesn't already have a hole in it from you people cutting holes in it. And so they gave us a bit of this horse skull, and it had teeth in it, and teeth are very diagnostic for horses, and it had the entire skull shape, and it's a gorgeous specimen horse. We extracted DNA from it, prepared it into a library and sequenced it, and then compared it on a computer to all living equids, and it's not a horse. It is a thing that paleontology has not named. It is slightly more closely related to donkeys, but it's not a donkey. It's about 4 million years diverged from donkey and about 4.5 million years diverged from horses. It's a new species, a new North American endemic species of equid that's somewhere closely related to horses and zebras and donkeys, but not one of them. And that is just an example of kind of how cool it is to be able to go into these places and get these new bones and just extract DNA and figure out what they are and learn about how species come and go and change and the habitats change. And you're always going to be surprised. It's a
Yeah, no, that's amazing. That's so cool. It does not sound like the kind of CV that you'd expect from someone who then goes and writes a book about like CRISPR and all this stuff about biotechnology. So
to be fair, though, that's the second half of the book. The first half of the book really is about ancient DNA and what we've learned using the genetics, the DNA that we've been able to extract things from the past about our species changing relationship with the plants and animals that we encountered as we evolved and spread across the planet and moved around. And it's those technological changes and innovations that bring us to where we are today, where it's very useful to start thinking about these new technologies and how they might be used going forward to help with the biodiversity crisis.
So you feel that in order to be thinking about these issues about CRISPR or about using these technologies to deal with food or the extinction crisis and so on, you think that it's valuable to go back to 50,000 years ago, to the start of humans manipulating nature?
Yeah, 50,000 years is kind of a random number that was nice and
Sure.
round and we can use, but yes, I do. And I think that's because there's a lot of people think that we are standing at the precipice of something that is different. We are just about to start messing with things. We have a decision to make about whether we're going to continue to stand off and just be humans and let stuff evolve, or we're going to really start to meddle and get our hands dirty. But what we see when we look at the past is that we have been doing this for as long as we've existed as a species. I mean, initially we were driving things to extinction, and this evidence is from the fossil record where people arrive in a place and you see mass extinctions of megafauna and other plants and animals. And some of this was from hunting and some of this was from habitat change. And in lots of places, there's also no climate change at the same time. And it's pretty clear that a lot of these extinctions are driven by us. That, of course, wasn't intentional. People didn't go to Australia for the first time and intend to kill off all the megafauna, or move to New Zealand and intend to kill off all the moas. But at some point, our relationship with these animals and plants did become intentional. And that is when we start seeing the first evidence of domesticates. And the earliest domesticates are dog, but the richest fossil record is really domestication of things like goats and sheep and cattle, and the early farming that we see somewhere between 10 and 15,000 years ago around the area of the Fertile Crescent. And what happens, I think, and you know, a lot of this is interpretation of the fossil record, of the archaeological record, but people realized at some point that keeping their family fed did not necessarily mean that they had to kill all of the aurochs that were out there. And we learned, our ancestors learned, that if you treat these populations slightly differently, if you tweak your hunting strategies, then the population survives. So if you only take males, or if you only take elderly females that are non-reproductive and leave those that can have offspring the next year, then your population is sustained. And this is the first evidence of deliberate manipulation of other species. And then of course we start bringing them closer to our settlements, and then our ancestors started choosing, you know, which animals were going to breed and which animals didn't, and making sure that the ones that spooked and ran away didn't contribute to the next generation. We started shaping the evolution of these species, and that was thousands of years ago. So
No, I was just going to ask, sort of tying back to what we were talking about before, you know, for a long time, you know, what we understood about that transition, you know, the beginning of agriculture, for example, was, you know, it was archaeologists like digging up, you know, seeds or pottery or what have you, you know, maybe some dog bones. And now, you know, we have this new set of kind of information that people like you can offer of DNA, either in those ancient remains or just looking at sort of, you know, DNA in living domesticated things. What do you think are some of the most important things we've learned about like what happens, or genetically speaking, when things started to get domesticated?
I think we're still really in the early stages of figuring this out. I think one thing that probably people suspected, but is clear from the genetics, is that, for example, with cattle domestication, we know that this probably didn't involve very many individuals initially, that they must have been quite hard to domesticate, these big aurochs, and it just took a few individuals that they were able to tame. Really, taming took much longer than this, but bring close to them and sort of direct their reproduction for some time. But then as cattle began to move around, they also hybridized with wild animals that they encountered. And people were probably doing this deliberately to introduce new genetics into their stocks, or to bring in different haplotypes. There's a haplotype in Tibet.
We should mention what a haplotype is.
Sorry. Yeah. So, you know, for every one of our genes, we have many different versions of those genes that are circulating among people. And those versions are sometimes called alleles or haplotypes. It's not that important to call them that. We can just say they're versions of genes, but there's a version of a gene that makes it easier for animals that are living at high altitude to survive in this low oxygen environment. And we now know that the domestic cattle that live in high altitude Tibet inherited that gene from hybridizing with local yak. We, people, inherited that gene from hybridizing with local Denisovans, and wolves and now domestic dogs, like Tibetan Mastiffs, inherited a version of that gene from an ancient lineage of wolf that doesn't exist anymore, that's just passed that down. So there are places in the world where it's tough to live, and some species have evolved some really cool adaptation to be able to do well there, and they pass that on to other things. And so domestication involved a few animals, but then it also involves moving genes around, and it involves deliberate selection by people that cause the animals to change and change shape, change their construct, change their physiology, change their behaviors by decisions that we make when we're not we, but our ancestors made when they're figuring out where, who's going to live, who's going to reproduce, who's going to be part of that next generation.
And I mean, are there particular kinds of genetic change that seem to happen during domestication, like, or during that kind of change? Like,
Yeah,
are there?
this is kind of controversial.
Mhm.
There's a syndrome that's been called domestication syndrome
Okay.
that involves a suite of changes that seem to have similar morphologies between different species. This piebald coloration, floppy ears, and curly tails. All of these seem to be associated with um, um, versions of genes that cause animals not to stress out in the presence of people. And selection for those has these secondary phenotypic effects where it changes the way these animals look. So pigs and dogs and cows and other things seem to all have these similar characteristics that they couldn't have inherited from the same ancestor because they're so divergently related. So it seems to be a consequence of domestication. In plants, we have similar things. Probably the trait that people were most interested in plants were plants that held on to their seeds for longer than other ones so they could be harvested all at the same time rather than have the seeds fall off and then go on the ground and start to rot or become buried and start to grow. And so there's been selection in lots of different plant species for this trait to hold on to your seeds that they can be harvested simultaneously. But you can imagine, yeah.
And yeah, sure. But is that like, should we be thinking about this as like one mutation that voila instantly creates a super domesticated crop, or like a series of mutations? How does that work?
Yeah, it's a great question. And it's most likely it's a series of lots of different things. I mean, we are for most species that aren't us and things that we grow in our labs, like fruit flies and little see-through worms, and we don't know very much about the genome. We don't know much at all about how the genes actually interact with each other to cause the way something looks or acts or yeah, exists, right?
Right.
And so probably these are suites of interacting genes, and we just over millennia were able to select for those that had the combinations of genes that produce the phenotypes that we were most interested in seeing move on to the next generation. This is really where the technologies that we have at our fingertips today start to differ from these traditional technologies of just blending two organisms together and hoping for what's best. Because now with gene editing tools, we can actually focus on a very specific trait and engineer that one gene to be different in the next generation, which is something that we haven't been able to do before. And this is important. I say that we've been manipulating the evolutionary trajectories of everything around us for as long as we've existed. And the new technologies are an extension. They're the next thing from what we've been doing, but they are different. And it would be dishonest to say that they weren't different. We've never before been able to manipulate things so quickly and precisely to be able to create the genetic differences that we want to see. But importantly, we also don't always know that those genetic differences are going to have the phenotypic consequences that we want, because for most species, we still don't understand that link between the DNA sequence and how something looks and acts.
Right. Just want to remind people, you can, as we're talking, you can leave questions for us, and then I'll try to get to as many of the questions as we can. Well, we're probably going to talk for maybe 10 or 15 more minutes, and then we'll do questions for your questions for the rest of the time. Where are we with this kind of new way? Let's focus on crops and just agriculture. The manipulation of crops and livestock has been, intensely controversial for, 30 years now, maybe. But that started with what was called, genetic modification, of GMOs. And now you're talking about gene editing like CRISPR. So I'm just very curious, maybe if, when you take a look at where the science is with making these manipulations to our food, where do we stand? I mean, is this still, kind of a nice idea in the lab, or do you see this as really very soon affecting what we're eating?
I think we're at a really important period where we are on the precipice of making some decisions that will impact this. And in the early days, before CRISPR, in order to make changes to a genome, you had to insert a bit of DNA from a different organism, a microbe normally, and use that as a way of making these changes. And now with CRISPR, we don't have to do that. And so now, so a lot of the objections that were early on were to this idea that we're combining DNA from across the tree of life, that we're creating something, and I don't think this is necessarily bad, but this is where the objections were coming from, creating something that wouldn't naturally be created. And today in the ag space, there's a difference in the regulatory pathway if we use our tools to create something that might have occurred in nature, given enough time, and something that couldn't have occurred in nature. And the difference here is in what we call cisgenic versus transgenic gene edited or genetically modified organisms. Trans just means from two different organisms, two different species, and they come together. So if you're transgenic, you have DNA from two different organisms. If you're cisgenic, then you have DNA just from the same organism. Maybe what's happened with CRISPR, we don't have to insert DNA. We can, we don't have to. What we might do, for example, is just turn a gene off or turn a gene up or turn a gene down. And in doing that, really affect the phenotype of the organism that's there. And there's some cool examples of this, but I started with the regulatory pathway, so I wanted to finish that. In the US, plants that are cisgenic, gene-edited plants, so they could have been created just by regular breeding in the field, but they weren't. They were created instead using CRISPR and the tools of gene editing, are not regulated. So you can have these things on the market, and we have a few of them now. The Arctic apples that are non-browning, there's a mushroom that does this. But if you're an animal though, even if you are cisgenic, you're regulated by the Food and Drug Administration. And just after the USDA, which regulates plants, decided that they would not regulate gene-edited cisgenic could have happened by nature sort of things, everyone was expecting that the FDA would make the same decision, but they didn't. Instead, these animals are regulated as drugs, which means that in order for an animal that has a cisgenic change, a gene turned off or a gene turned up or down, to get onto the market, it has to go through the same type of trials, phase 1, 2, 3 trials that we're all very familiar with now because of coronavirus, that a new vaccine or a new cancer drug has to go through, which really limits the type of people, type of organization that can bring one of these products to market. It's the regulatory pathway is really inhibitory at the moment from what I think could be really, really good for agriculture and also for conservation. We said a minute ago, there are some cool things. There was a tomato that was just, I think, deregulated or put into market in Japan, where the environment for accepting gene-edited organisms is different than here. And what they've done is turned off a gene. So they just went into the tomato genome and added a stop in the middle of a reading frame for a gene. So the gene stops making copies of itself. And what that gene normally does is determines how much of a protein is made in that tomato and then sort of stops it after it gets to a certain mark. It's auto-regulatory. But by stopping it, that protein actually increases in abundance and continues to increase in abundance. And the protein that's being made is the GABA protein, which is associated with lowering heart pressure. So it's kind of a good thing to have lots of GABA protein. Tomatoes are good if you have high blood pressure because they make this protein. And here are these gene-edited tomatoes that just have this one mutation, stops a gene from making something that gives you something like 7 times the amount of this GABA protein that normal tomatoes have. And this product, as far as I can see from the news, has been embraced by the market because it's an exciting thing. It doesn't have genes from anything else. It's just tomato genes. It doesn't have any extra genes at all. It just has one gene turned off, whose job it is to sort of regulate the amount of that protein that's produced. And these are the types of innovations that could be possible with these technologies if we allow it to be there.
And I mean, what is possible? How is it, how do I put this? Why do we need to do these sorts of things to the things we eat, as opposed to more traditional ways of altering the DNA of animals and plants that we consume?
Yeah,
I mean,
the challenge that we're facing is that there are too many of us. and that the quality and quantity of arable land on the planet is actually less than it was decades ago. And there are many more of us that we have to feed. We don't have to embrace these technologies, but if we don't, there are people who are going to starve because we're not going to be able to make enough food to feed them. And this is, even if you're opposed to transgenics, and I'm not opposed to transgenics, but if you're opposed to transgenics, you can think of this as a way of just speeding up what we could eventually get to anyway, but getting there faster because we now understand the way these different genes interact with each other, and we have the ability to make the very specific change we want. For years we've been using in agriculture this strategy called mutation breeding, where we take a plant that we're interested in making new versions of or new varieties of this plant, and we plant it and we zap it with radioactivity or with chemical mutagens and cause tens of thousands of mutations to form across the genome that are never characterized. We don't care about those at all, right? And we look for varieties that are interesting to us in some way, and we pick those up and we propagate them, and if they survive and they thrive, then we're like, that's a great product. And that product has tens of thousands of mutations that are completely uncharacterized. Mutations are not inherently dangerous, right? So this is not, I'm not saying that we shouldn't use mutation breeding. I'm just trying to point out the fallacy between being scared of making a very specific change, but being completely accepting of things like ruby red grapefruits and Redefined wheat and brown rice and other things that we don't think of as genetically modified at all, but are way more human modified than some of these gene edited things that we're thinking about bringing to market.
So you mentioned conservation, and this is something I've found very intriguing. Stimulating, but perhaps worrying at the same time is using these tools to try to protect endangered species by altering them. Maybe you could talk a little bit about, maybe we could talk about an example. I mean, what's your favorite example of how this might be applied, just to give people a sense of what on earth we're talking about? Because it's just,
sure,
we're so used to thinking about, manipulating the things within our orbit, and wildlife is out there. We don't mess with that, but that's what we're talking about.
And in some ways, because of this is harder. We can manipulate corn and wheat and rice because we know a lot about their genomes. We have a lot of experience manipulating them. But in some ways, the things that are most desperate for this sort of innovation are those species that we don't know very much about, that are really on the brink of becoming extinct because their habitats have changed to such an extent that natural selection, natural evolution is not going to keep up. My favorite example actually is the black-footed ferret at the moment. So this is the black-footed ferret was one of the first species, one of the original listed species, the 1967 Endangered Species Act, the first list. And at the time, there was a single population that was left, and there was tons of effort that was thrown to creating a captive breeding colony of black-footed ferrets, and it failed. They couldn't figure out how to make the black-footed ferrets breed in captivity. They live across the North American plains, and they eat prairie dogs. So they live kind of the same place where prairie dogs live. And prairie dogs are annoying if you're a farmer or you're building cities or roads. And so throughout the 20th century, there were these very successful prairie dog eradication programs. And these programs also eradicated the main predator of the prairie dog, the black-footed ferret. So by the 70s, I think it was, they thought they were entirely extinct. They didn't know for sure. They stayed on the list. And then in the mid 80s, a single population was discovered near Meeteetse, Wyoming. And this gave US Fish and Wildlife and other local stakeholders a second chance to try to figure out how to make it work. And this time they knew a little bit more. They brought in people who were experts in rearing different types of wild animals in captive environments, and it worked. They were able to establish a really successful captive breeding program for black-footed ferrets. And every year, about 500 black-footed ferrets are released into the wild, and some of them do okay, but most of them don't. And there are two problems that are plaguing black-footed ferrets. The first is that this one population that was left was just one residual population, and they are really suffering from the effects of inbreeding. In fact, there are only 7 founders, and these founders are extremely closely related to each other, to the entire captive breeding program. So they don't have very much diversity, and it's very hard to mitigate against just all of these deleterious effects of everybody being closely related to each other. The second and kind of more insidious problem is that sylvatic plague, which was introduced from Europe, is really common across their range.
Is this like, was this like
the
bubonic
plague? It's the plague. Yes.
It is the plague. Okay.
And so prairie dogs are commonly infected. Black-footed ferrets eat a prairie dog, they get the plague and they die. You can vaccinate them, and then you can capture them and revaccinate them. And in the wild population, you have to capture them a couple of times and vaccinate them, but this is not a sustainable conservation program. But there are solutions that come from these new technologies that we have. The solution to the inbreeding problem actually has potentially just been solved, or perhaps at least solved a little bit. In San Diego, as part of the San Diego Zoo, there's a resource facility called the Frozen Zoo, where people, scientists, have been collecting samples of animals that are endangered or destined to become endangered or extinct, and putting them in deep freeze so that you have living tissues that can be brought back to life and then regrown, as it were. And they have a couple of black-footed ferrets in that population that weren't from this population in Atitzi. They were from the population that was part of the original captive breeding program that failed. And about a year ago, an animal, her name is Elizabeth Ann, was born. She was cloned from one of these tissue cultures that was in the deep freeze at the San Diego Zoo for more than 30 years. And she's perfectly healthy, happy black-footed ferret. She will become the 8th founder lineage of the captive breeding program once she reaches reproductive maturity. And she's not related to the individuals that are in the captive breeding program now. So she will bring a welcome bit of genetic diversity to this population. But she's not likely to solve the problem of plague. To that, we have to turn to these new technologies and to transgenics rather than thinking just about cisgenics. There is a closely related species of ferret, an evolutionary cousin, if you will, of the black-footed ferret called the domestic ferret. You know, we all know domestic ferrets as little stinky things, like kind of cute, not as cute as the black-footed ferret. They do smell weird, right? Have you smelled a ferret? They do smell weird.
They do. I don't know how black-footed ferrets smell, but
me either.
Yeah.
Actually, fair enough. They might also smell.
Who
Yeah,
knows?
they probably do. I mean, it's a ferret, right? But they're cute. They're super cute. Look up
They are
Elizabeth
cute.
Ann. She's adorable. So domestic ferrets, though, evolved in Europe alongside plague and have natural immunity to it. They're exposed to plague, they don't get it. And so Revive and Restore, which is a nonprofit based in Sausalito, California, in collaboration with San Diego Frozen Zoo, US Fish and Wildlife, the Smithsonian, and a whole bunch of academic and governmental and NGO partners have been working to try to figure out what it is about the genome of the domestic ferret that makes them not get plague, with the idea that once they can figure this out, this is a hard question, but once they can figure it out, they can use gene editing, genetic engineering technologies to transfer that bit of genetic resistance from the domestic ferrets to the captively bred black-footed ferret. So you'll have a transgenic black-footed ferret that is mostly black-footed ferret, but a tiny little bit, maybe, domestic ferret, but a ferret that is capable of...
But just to clarify, we're not talking about taking like some adult black-footed ferret and just inserting bits of DNA into its cells. We're talking about something reproductive.
Right. This is a perfect example of why just breeding two things together using the traditional style wouldn't work, right? So if we bred a domestic ferret with a black-footed ferret, that might work. It might transmit the genetic resistance to plague, but it would also probably transmit, a penchant for liking cuddles and, not really being able to survive in the wild as a black-footed ferret. We don't want half of the genome to be a domestic ferret and half of the genome to be a black-footed ferret. We want the whole thing to be a black-footed ferret, except for that domestic ferret's bit of evolved resistance to plague. So we need to use these technologies that we have to transfer just that little bit of DNA.
And transfer that to
the black-footed ferret, to an embryo.
To an embryo, okay.
Yeah, and then have that embryo become part of the captive breeding pool. It sounds crazy. And, from the perspective of this is a wild animal, we don't really know what gene is causing this trait. Is this an absolutely insane thing? Maybe it is kind of crazy, but it does give us hope. And there actually is one species, one so far, where this has worked, where there is a wild species that is in danger of becoming extinct, that has had a little bit of DNA from a different species transferred to its genome. And that species is now on the verge of a comeback. Do you know what that is?
Do tell.
You do know though, right? It's the American chestnut tree.
Of course,
Right.
right.
So this is a tree that used to make up the bulk of the forests across the East Coast. You must have written about this. You must have written about it in your National Geographic article.
Yes, I was so fixated on animals, I forgot about wild plants. Yes, but yeah, the American chestnut, the
This
poor
is
thing,
such a
that
great story,
a
right?
magnificent tree that got crushed by
By a fungus.
By a
Yes.
fungus, yeah.
That was introduced from Europe. And what that fungus does is it burns holes through the trees so that the fungus can spread through those trees. And then it, sporulates when it rains. And it causes the tree to leak all this gross stuff and have a little, like a belt forms across it, and then it dies from above and dies from below, right? But what Bill Powell and Chuck Maynard realized was that all they needed to do was find a way to neutralize that fungus, and that lots of plants can do this. And so they turned to wheat, and they could have picked lots of things. They picked wheat probably because we eat a lot of wheat, and so if we eat a tiny little bit of wheat DNA and some American chestnuts, we kind of know what's going to happen there. But wheat lives with this fungus. It makes an enzyme that reduces the acidity that the fungus produces and causes the wheat not to die. And so they created transgenic American chestnut trees that have a tiny little gene from wheat that makes exactly this enzyme that neutralizes the acid of the fungus, and the American chestnut trees can survive even with the fungus. So, and they're in the process of going through regulatory approval by lots of different regulatory agencies at the moment. But these trees exist. There's a lineage called the Darling line, the Darling variety of American chestnut trees that my name is on a list to buy as soon as they become deregulated. So, and this could be the beginning of reestablishing what was once the dominant tree in the Eastern American forests.
What about this thing called gene drive? People may have heard of this. I mean, this is this is as I understand it, is a way to basically like introduce genes into a population at top speed. I mean, maybe you could talk a little bit about how that works and why you would want to do that.
Yeah.
And
so,
what your view is on it. Period.
Yeah, gene drives are a little bit trickier. They're a little bit trickier to explain, and kind of trickier to think about how you feel about introducing them into the environment as well. But so what happens is normally when you inherit a version of a gene from your mom, everybody has 2 chromosomes, right? You get one from your mom and one from your dad. And normally if you inherit one version of your gene from your mom, you have a different version from your dad. And if you were to edit mom's DNA and then make this offspring, you would get the edited version just from mom and the unedited version from dad. What the gene drive does is it puts all of the editing machinery, all the stuff that you need to actually do the genetic manipulation, the CRISPR machinery, along with mom's gene. So when you inherit that gene from mom, the first thing that happens is that editing machinery makes copies of itself, goes over to dad's chromosome and edits dad's chromosome. So you inherit a version from mom and a version from dad, but dad's version quickly gets replaced by the version from mom. That doesn't normally happen in evolution, right? So the gene drive is driving that gene, mom's gene, through the population at a pace that is way faster than evolution would allow. So what people are thinking of doing with this is, for example, using it to make things sterile. So if I wanted to get rid of invasive rodents on an island, for example, I might introduce a couple of edited rodents that have a gene that makes them sterile and a gene drive that means that once a egg is fertilized with their sperm or whatever, that individual will also have that sterility gene on both copies of both chromosomes, both copies of that gene. So the idea is that within just a few generations, that sterility gene or whatever it is that you're trying to change will have passed through the entire population and it will go extinct, right? So people are thinking of using this to get rid of invasive rodents on islands, and also to get rid of things like mosquitoes in places where mosquitoes are really important in causing human disease, things like dengue and chikungunya and malaria. The problem is it's a scary thought to think that you might be really changing everything that quickly and have no control over what happens. And there are no gene drives that are existing in the wild right now. Right now, people are just having conversations about how you might do this. But everyone who's been talking about this is also really adamant that we need kill switches in these gene drives, or ways to make sure that they don't last forever. And people have invented a bunch of different ways to do this, either by introducing another drive that actually reverses the effects of the drive that are there, or by having drives linked together in what they call a daisy chain in such a way that the end of that chain is lost in every generation. And so the effects of that gene drive can only last for as long as the number of links in that chain, until it eventually just goes back to normal. And this is a way potentially of suppressing, for example, mosquito populations for some number of generations before you have to go back in and do it again. And this is a huge improvement over going in and just spraying chemicals every couple of months or something. You could release these gene edited mosquitoes that have a drive element that only lasts for, say, 10 generations before it disappears. And you only have to insert them once, and then you have a couple of years before you have to go in and do it again. And in the meantime, you suppress the whole population without having to spray with a whole bunch of chemicals or pesticides. So there are thoughts that this is going to be a really exciting way to do this, but it's still very much in the early stages of really talking about the ultimate consequences of this with all of the stakeholders who might be impacted. And start thinking about things like this. It's really clear that we can't make a decision that's going to impact just one community, that everything is linked on our planet. And if we're going to start doing stuff like this, we really need to have global conversations with stakeholders from all parts of the world who are involved in all aspects of the way that we interact with the planet to make decisions like this, which is another reason why it's important to have these conversations out loud, rather than to just have some companies trying to figure out what's going on in the background without telling anybody.
Let's go to some of these questions. So one question is, can our meddling technologies help prevent the next pandemic?
It's a good question. One of the ways that we could try to prevent the next pandemic is to not be encroaching on wild animals to the extent that we are. So coming up with ways that we can better use the land that's already been cleared, that we can make more food with the space that we already have without having to go into forests that have come down, really to minimize the contact with other species. And I think there are ways that we could use our meddling technologies to do that. But often when people ask this question, what they mean is, can we engineer ourselves so that we're resistant to a disease, or can we engineer the disease so that it's not infectious to us? And unfortunately, that's too hard. The only thing that we can do as engineers is the same thing that evolution does. We try out some things, and sometimes it works and sometimes it doesn't. And whatever the generation that is born, those that in that generation that are the most fit evolutionarily are just the ones that can survive in that landscape. And as soon as we tweak a pathogen or our own genomes in some way, the pathogen will evolve another way to get to us, and that's really what would happen. I mean, this is, also one of the reasons that one of the scariest people think about, one of the scariest things about these editing technologies is, what if we edit ourselves? What if we use this to create some super fit, super amazing person, and that just increases the inequities that we already know that separate people in this world? And I also don't think that would work, because that's us trying to imagine that the thing that is super awesome today is also going to be the thing that's super awesome in the future. And again, that's just not how evolution works. As soon as we determine what's super cool, it's mom jeans, or it's neon, or it's whatever like my generation was wearing when we were in high school. Like, that's not cool anymore. So we could try to imagine how to design super fit people, but that's just not, or pathogens that are somehow less infectious, but that's not really, I don't think that's really how we can use our technologies.
Gotcha. Going back in time, do we have an idea of what the first animals that became extinct were as a result of human intervention? So what did we drive into oblivion first?
I think the archaeological record suggests that the first megafaunal extinctions outside of Africa, and we have a much poorer record of what's happening in Africa, because of course there's a very long history of humans living in Africa alongside animals, and also extinctions that are tied both to probably periods of human expansion and also rapid climate change. But once people left Africa, the earliest archaeological evidence of humans turning up in a place, and also megafaunal extinctions is in Australia. So this is the first place that the earliest wave of people got to, and there is very strong evidence of simultaneous wave of extinctions. And Australia is also important for another reason, and that is that if we look at the timing of humans appearing in Eurasia and in North America and South America, we see coincident extinctions of megafauna with the first people, but this is also a time of really rapid climate change. And so there's been this tension between what caused it. Is it people, or is it climate change, or is it both? And in these places, I think it probably is both. But if we look at somewhere like Australia, there's tons of evidence of climate change before people are there and after people are there, but no evidence of mass extinctions during the old periods of climate change, and tons of evidence of mass extinction with the first appearance of humans, and no coincident climate change. And the same is true with the most recent extinctions, which happen mostly on islands, where we see people arriving much later on islands. There's no longer any periods of rapid climate change, but again, we see this coincident timing between the first archaeological evidence of people and megafaunal mass extinctions, things like the moa and the dodo. Yes.
And in Australia, what was there? I mean, there weren't
giant wombats and giant kangaroos and all sorts of
how
cool
giant
things.
are we talking about?
Oh, you got me. I don't actually know.
Oh, okay.
I think I read they're about the same size as late Pleistocene mammoths, so really big wombats.
Cool.
Yeah.
I'm sorry they're gone.
Me too. Yeah.
So I know someone else asked, what are some of your biggest concerns about CRISPR? So we've been talking about wonderful potential in all sorts of ways. What are you worried about in terms of CRISPR?
I think that's a great question. I think it's hard to know yet what to be most worried about. I think we're still very early in trying to figure out what we can do with CRISPR. What I'm most worried about right now doesn't really have to do with the CRISPR machinery itself, but in our real lack of understanding about what happens when we start to change genes. We for most species, like I've said, we really don't understand the link between what the DNA sequence is doing and what that means about the way an animal looks or acts. And that's going to take a long time to figure this out. And I guess I'm a little bit worried that there's going to be too much hype and excitement, and the advances aren't going to be fast enough just because we're missing some of this fundamental biological knowledge that there's kind of an expectation that we have, and yet we don't. Like the black-footed ferret example, we could walk around and say, we're going to make black-footed ferrets that can't get plague because domestic ferrets don't. We're going to use CRISPR to do this. But we have no idea why domestic ferrets don't get plague. And it might take us years to figure that out. And in the meantime, are people going to be like, well, you said CRISPR was going to solve this problem? Well, CRISPR is ready. It's us, the biologists, who don't know what to do yet. Mm-hmm. The genetics biologists trying to figure out what to do.
Right. I see. Another person asks, how much are scientists cloning today? It used to be big news item, but sounds like it might not be anymore. And I certainly remember, in the 70s, it was there was this idea that you could clone people. And then, Dolly the sheep was actually a cloned animal in the 90s. Right. And then there would be some I remember I don't know if you saw, there were these weird reports, somebody claimed to have cloned babies, but wouldn't actually show you the babies, or I mean, it was there were these weird reports, but but cloning is not really something in the headlines these days. So what happened?
It is common in agriculture. It's pretty inefficient. There's a step in the cloning process so cloning involves taking a somatic cell. So our bodies have basically two types of cells: somatic cells, which are mostly all of them, and germ cells, which are sperm and eggs. And normally what happens is a sperm fertilizes an egg, and then you have both copies of the chromosomes, and then that starts the process of beginning to divide and differentiate and become all the different cell types that make up an animal. In cloning, what happens is you take some somatic cell. In Dolly, it was a mammary cell, but a lot of times now people are using skin cells or other types of cells. It could be any of them. And you take that cell and you stress it out, grow it in a dish in the lab and sort of stress it out. And you can harvest an egg cell that's ready. Then you suck the nucleus out of that egg cell. So that would have been the copy, the chromosomal copy that comes from mom, but you just get rid of it. So now you have an egg cell that's all primed and ready to go, but it's missing any DNA whatsoever. And you take this stressed out somatic cell, put it next to the egg cell, zap it with a bit of electricity, the cell wall opens up because of that, and the nucleus from this somatic cell dumps into that egg cell. So now you have essentially what would have been a fertilized egg cell, 2 copies of the chromosome, but both copies have come from the donor of whatever somatic cell it was. But then comes the really hard step. So that somatic cell is very good at being a mammary cell or a skin cell. You don't need the same genes to be on or off or up or down at a particular extent to be a mammary cell as you do to be a brain cell or a heart cell or a skin cell. So you have to now trick that cell to forgetting all of the instructions to being the type of cell that it's programmed to be and reverting to that type of cell that you have when you just have a sperm fertilizing an egg. And so that happens normally by, again, maybe another electrical current, and you zap that egg, and the proteins that are in the outside of that egg cell cause it to forget all of its instructions and revert to that stage. This is super inefficient. And even with domestic animals that we know how to do this, it's a super inefficient process, but we're getting better. I mean, we can clone sheep, and there's a company in China that announced a few years ago that they were going to create 500 clones a month of Wagyu cattle to satisfy the beef market because they had this perfect animal that they just wanted to make immediate exact copies of. So, it is common in agriculture, but it's hard to get the right set of experimental conditions to make this step where you trick that cell into forgetting its job and reverting to this primordial type of cell. It's hard to get those conditions for a lot of animals. And also, we don't know a lot about reproductive, what do we do? Okay, mammoth cloning. Everybody wants to talk about mammoth cloning at some point. There was even a company that was formed a couple weeks ago to do this called Colossal, right? So they might be able to take an Asian elephant cell that they grow in a dish in a lab and use gene editing to cut out all the bits that make it look like Asian elephants and replace it with mammoth bits. Impossible now, but technical hurdle. Maybe at some point they can make 1.5 million edits to a single cell and have a mammoth genome in an Asian elephant cell. Then you have a living cell that you could clone if you could get it to forget all of its instructions and revert to the cell. But let's say that works, right? Then you have this developing embryo of an mammoth, whatever, and you got to figure out what to do with it. Can you just put it in a surrogate elephant mom host and hope that it all goes well? A lot of the instructions that happen when a fetus is developing actually come from the mom, not from the fetus itself. So that's going to be elephant instructions. And in elephants, we don't even know how to do IVF. Like we don't know how to do any of this stuff. So we don't hear about cloning a lot for animals that we don't work with a lot, just because there's so many technological hurdles that need to be overcome in order to make this happen.
Yeah, and my understanding is that the plan is to raise these mammoth embryos in bags, basically, like artificial
womb. Yes, this is the plan. Those don't exist yet either, though. So yeah.
Right. Well, okay. This is you sort of preempted the last question I was going to ask, but I'm going to ask it anyway because I like the question. So the question is, and you can take this any direction you want, will we actually clone a dinosaur one day?
Ooh. Well, we have birds. We could probably clone birds. Those are dinosaurs, right? That's not the way you wanted me to take it. No. Okay. So
they are birds, but we haven't cloned birds yet, right?
No. And in fact, it's really hard. And the problem there is, again, technical. Well, let me answer the dinosaur question first. And non-avian dinosaurs. And then we could talk about briefly the challenges of cloning birds. So non-avian dinosaurs went extinct more than 65 million years ago. A few months ago, we broke the record for the oldest DNA ever published by getting DNA from a mammoth tooth that's around 1.2 million years old. This is way breaking the record for the oldest DNA, but it is a far cry from 65 million years. So there is no chance that we are going to be able to get DNA from a non-avian dinosaur. It's just these bones are rocks, and rocks don't have DNA. They're cool, right? And there are some of them that, Mary Schweitzer, her group at NC State, sometimes they isolate these things in slides that have me convinced. I'm looking at it and I'm like, holy cow, that looks like an actual cell with antibodies that you can stain, et cetera. And she sends them to me and I grind them up and extract DNA and get nothing as completely expected, right? We have this back and forth relationship too. We're not going to get any DNA from dinosaurs. So we're not going to be able to clone.
Sounds like a dysfunctional relationship. You just keep breaking her heart.
I think we came up with this plan over drinks one day at a meeting many years ago.
Sounds like much alcohol was involved in this plan. Yeah.
She's convinced that someday she's going to send me some that work, and I am willing to give it a shot. Right?
All right.
Yeah.
Okay. Yeah. Okay. So dinosaurs, super skeptical. These bones are rocks.
Birds. This is a cool thing. And I think one of the, Colossal, I think, is a great idea, right? Because
this is the Mammoth Company.
The Mammoth Company.
Yeah.
They're gonna develop new technologies that we can use for conservation, and I am hopeful, extremely hopeful. In fact, kind of forcing it in conversations with the founders, Ben Lamb and George Church, that this is one of the directions that they're willing to go. Figure out how to clone birds. Birds are in trouble everywhere, and even if we could gene edit them, we have to be able to take those edited organisms and edited cells, edited DNA, and turn it into an actual living, breathing bird if it's going to have any impact. And the challenge with birds is that you can't get the egg at the same developmental time that you can from something like a sheep or a cow, where you can go in and you can give the female hormones and you can harvest a whole bunch of eggs at once, and you know that these eggs are ready to go. That doesn't happen with a bird. With a chicken, for example, the egg becomes ready to be fertilized on insemination, and just as that sperm is about to go in, so you can't get it. By the time a chicken lays an egg, there's a 24-hour-old developing embryo in there, and so you can't rip that out of the thing and edit it and put something else in there. It's way too many cells for that. What you can do, though, is at that stage, the primordial germ cells, the cells that will eventually become either sperm or eggs, but aren't yet because the gonads don't exist yet, so they can't be there, are migrating around the outside of that embryo, like in the egg. You could stick a needle in that
inside
egg.
Of the egg.
Inside
of the egg, these
developing embryo,
sperm and egg
cells moving around outside the embryo. You can stick a needle in there and suck those cells out.
Okay.
Yeah. And you could put them into a dish. You could edit them. Yeah. And
then what?
Then the next egg that you have, you could shoot those edited primordial germ cells into that animal, and that animal will grow up and be perfectly normal, except some of their eggs or some of their sperm cells will be edited, right? So many years ago, not that many years ago, but some years ago, we know this is true because somebody, I don't remember who, injected duck primordial germ cells into a chicken egg. And that chicken grew up and was perfectly happy and healthy. And then that chicken was fertilized with duck sperm. And the duck sperm only fertilized the duck eggs that were inside that chicken's gonads because they'd been injected in there with a needle. And she laid a bunch of eggs that hatched into perfectly normal ducks. So the plan is to use that, but to create gene-edited birds. Can't do that yet, but it would be super cool, and it would be super awesome for conservation because there's tons of birds. Think about the birds in Hawaii that are just really holding on because of avian malaria. Like if we could figure out how to make those birds resistant to malaria or other birds resistant to the diseases on islands that are killing them, then that would be a fantastic advance for conservation thanks to these new tools.
All right. So yeah, so let's work on saving our feathered dinosaurs that we have all around us. Great. Well, Beth, it's been great talking to you. I feel like we kind of scraped the surface of life as we made it. I mean, there's a lot more in this book for people to discover. I highly recommend that you dive in. It's great stuff. And yeah, good talking to you.
Thanks, Carl. It was a lot of fun. As always, I enjoy talking to you.
Likewise.
Well, on behalf of Town Hall, I want to thank you so much, both of you, for being here and presenting with us. You've presented so many things that I had no idea existed, and it really makes me feel like I need to pay attention to science news a little bit more. I want to thank the audience as well. Thanks so much for your questions. If you're interested in purchasing a copy of the book, I encourage you to use the link in the chat that's going to take you to Third Place Books, and we can support a local store tonight. Thank you so much, Carl, for your moderation, and Beth, thank you so much for juggling all that you're juggling to be here tonight. It's really awesome to see. So thank you so much, and best to you on the rest of your tour.
Thank you.